Hey everybody, we're back. Here we are finishing up the part 2 of mitosis. And that darn keynote just would not hold out for me to get in the rest of the slides. So let's pick up where we left off with cancer. Yuck. Hate cancer. Cancer is essentially a disease of mitosis. It occurs when basically cell mitosis goes awry. Cancer begins when a single cell is transformed or converted from a normal cell to a cancer cell. All the normal checkpoints that normally regulate mitosis are ignored or overridden by the cancer cell. And we started to discuss P53. P53, if you know any sort of tumor gene, this is the one to know. The normal P53 gene is the guardian of the genome. It usually functions to control all the proper cell cycle. This is the normal P53 gene is a tumor suppressor gene. But when P53 is mutated or abnormal, it does not suppress tumors. And it has been found that in over 50% of all human cancers, there is a mutated P53. And I was going to go into the more complex information about that. But with this sort of truncated or shortened term, we just don't have time. So I think that we'll leave it right there for you. Cancer cells have a number of unusual features. First, they're immortal. As we already said, normal human cells can go through about 50 cell divisions and then they die. But cancer cells can divide indefinitely, just like Henrietta Lacks' cervical cancer cells. Cancer cells also often have unusual numbers of chromosomes. Instead of the 23 chromosome pairs of a normal human cell, they might have, some odd, strange number of chromosomes. Another thing that happens with cancer cells is that they often have problems with their cell surface. Instead of sticking or adhering to its neighboring cells, the cancer cells will tend to round up like those in the petri dish we did in the last section, and they can break away attachments from the neighboring cells. And when they break away, they can seed elsewhere in the body, and then you have a metastasis. So what drives cancer cells to grow and divide uncontrollably and turn into cancer? And we end up having mutations in two basic classes of genes. Proto-oncogenes, which can then mutate and become oncogenes or cancer-causing genes, and tumor suppressor genes that when they're abnormal do not any longer suppress tumors. So proto-oncogenes are a group of genes that cause normal cells to be cancerous when these genes are mutated. Proto-oncogenes, when they're normal, will encode proteins that stimulate cell division and inhibit cell differentiation and halt cell death. Mutations in proto-oncogenes are typically dominant in nature. A proto-oncogene that has acquired a tumor-causing mutation has now become an oncogene. And oncogenes exhibit increased production of these proteins that increase cell division to the point of having uncontrolled cell division and decrease cell differentiation. It will also inhibit cell death. These are features that define cancer cells. Oncogenes are currently a major molecular target for anti-cancer drug design. Unlike benign neoplasms, malignant neoplasms progressively worsen and threaten health. A malignant neoplasm will divide abnormally, with populations reaching extremely high densities. Malignant neoplasms have altered cytoplasm and plasma membranes due to cellular malfunction. They have abnormal chromosome numbers, some present in multiple copies, others might be missing or damaged. Altered or missing proteins will impair the plasma membranes of malignant cells. We already discussed how they might not adhere well to their neighboring cells. So these cells might not stay anchored properly in tissues. And because their plasma membrane adhesion proteins are defective or missing, these malignant cells can slip easily into and out of vessels of the circulatory or lymphatic systems. By migrating through these vessels, the cells can seed or establish neoplasms elsewhere in the body. And this is the process by which malignant cells break loose from their home tissue and invade other parts of the body. Again, this is called metastasis. So cancer cells can invade by metastasis. They can also invade locally. So for instance, if you had a gynecological cancer, say a cancer of the uterus, it can just invade into nearby tissue, say for instance, into the colon. Same thing could happen with a prostate cancer that could invade into the bladder. So cancer can spread by metastases or by local invasion. And here is a nice slide that comes from your textbook that shows how the process of, in the first slide up there, number 1, how a benign neoplasm can grow slowly and stay anchored in home tissue. This could be like a wart, for instance. And number 2, cells of a malignant neoplasm can grow quickly and can break away from the home tissue. And then in number 3, we see what happens with metastases, when malignant cells become attached to the wall of a lymph vessel or blood vessel. And they release digestive enzymes that create an opening into the wall and then enter the vessel. And the cells will move through the vessel and exit the same way they got in. Okay, so let's do some practice questions. What is true of HeLa cells? Were they the first human cells grown in a lab that were naturally immortal, meaning that they do not die after a set number of cell divisions? Are they a cell line that was found to be remarkably durable and prolific? Were they the first cells put into mass production as cell factories, producing important products such as the polio vaccine? Were they the first human cells that were successfully cloned? And are they in use in labs throughout the world today, with scientists having grown an estimated 50 million metric tons of HeLa cells and almost 11,000 patents having been issued involving these cells? Well, you know the answer here. The answer is, of course, all of the above. This was one of those questions that really isn't a question. It's just more of a review slide. Mitosis occurs when? Is it when the cell divides completely? Is it in the cell cycle when replicated chromosomes are separated into 2 new nuclei? Is it in the cell cycle when the cytoplasm and organelles divide into 2 new cells containing roughly equal shares of these cellular components? Or is it when programmed cell death occurs? And mitosis, by definition, is when the cell cycle, when the chromosomes are replicated and then they are separated into 2 new nuclei. Mitosis is nuclear division. Cytokinesis is A and C, when all the rest of the cytoplasm and the organelles divide. And programmed cell death is apoptosis. During what stage of the cell cycle are the chromosomes copied? M, D, gap 1, gap 2, or S? And of course, the chromosomes are copied during the synthesis phase. Which of the following is the proper sequence for mitosis? And we have metaphase, telophase, prophase, and anaphase, and we have to put it in the proper order. And remember, I gave you a mnemonic device for that. And the mnemonic device, could I talk, is PMAT. So PMAT is prophase 3, metaphase, anaphase, and telophase. So that is the answer C. Chromosomes are aligned midway between the spindle poles during which phase? And this is so characteristic of this phase. It's really the only one thing you look for in this phase. And the answer is metaphase. So that would be when everything lines up on the metaphase plate. Are cancer cells, do they result from renegade cells escaping the controls on cell division? For sure, that's true, you know that. And what facilitates cytokinesis in animal cells that cannot work with plant cells because stiff cell walls surround their what? And this is about that contractile ring. The contractile ring that facilitates cytokinesis in animal cells, that's right under the cell membrane there, cannot work with plant cells because they have a stiff cell wall that surrounds their plasma membrane. And we're not really focusing on plants, but that's just one difference you should know. Dolly's lifespan might have been extended by giving her supplementary what? Dolly needs the fountain of youth, right? So Dolly's lifespan might have been extended by giving her supplementary telomerase. Now, in this next question, you need to know how to differentiate homologous chromosomes. And homologous chromosomes have which of the following features regarding length, genes, and shape? And homologous chromosomes have the same length, shape, and genes. Now let's talk about the chromatids. The chromatids detach from one another during which phase? And that would be anaphase. That's when they get pulled to the opposite poles. When cells are not responding to normal controls over growth and cell division, they form a tissue mass known as a what? And this would be a tumor. Tumors are solid tissue masses. Metastases are spread through the lymphatics or the blood vessels. Let's see. And let's go on to the last slide, which is the cell in this figure is in which phase? Okay, so you see we've already gone to the opposite poles, and doesn't it look like there's a furrow forming up above? So the cell in this figure is in telophase. The cell in panel 1 in the figure is in which phase? Okay, and none of the chromosomes are condensed in this phase, right? So this would be the cells are in interphase when all the chromatin is not condensed. The cell in panel 5 is in which phase? And here you see everything being pulled toward the opposite pole. So this would be anaphase. The cell in panel 3 is in which phase? And this would be prophase, but it would be later prophase. Panel 2 would be early prophase. Okay, now if we were having lab today, we would be looking at all these phases of mitosis under our microscope. Remember our microscope? It's been a whole month. Okay, so there's what your microscope looks like. And remember, you would get your microscope and you'd put your slide. We'd cut off little pieces of the onion tip. We'd stain the onion tip, and then we'd put the slide on the stage and start first looking for it under the lowest power, and then go up probably to the 10x. We'd find the pink stained area on the slide. You'd move the slide around, right, with your stage controls. And then we switch over to our 40x objective lens and adjust our fine focus. And we'd see something that looks like the next slide. So here, looking under the slide, we try to recognize our different stages of mitosis. And we get something that looks like the first stage in some slides, which would be early prophase. And then we have late prophase, or what could be called pre-pro-metaphase. Okay, so we're starting to get some spindles attaching to our centromeres. And then in the third slide, we see everything really quite dense and coiled up. And this is metaphase, where everything is lining up in the center of the cell at the metaphase plate. And then in the fourth picture, you see anaphase, which is the most condensed and coiled stage. And everything is moved over to the opposite pole, and you see the spindle almost like a little array of microtubules coming out of it. And then the last phase is telophase, and you can see we're starting to have a cleavage furrow and getting ready for the, almost getting ready there for cytokinesis, for the cytoplasm to divide. And this is just a reminder of where everything fits back in the cell cycle to put it all together. Okay, so we had all the stages of interphase that started with the gap 1 phase. Okay, and this is where all the cellular contents, excluding the chromosomes, are duplicated in preparation for that synthesis phase. It can also go off to that quiescent G0 phase, right, for cell cycle arrest. These would be like terminally differentiated cells, maybe like a nerve cell was our classic example. The S phase comes right after the gap 1 phase. And this is where each of the chromosomes is duplicated. So we make our sister chromatids at this stage, right? We had 2 homologous chromosomes, but each chromosome was only 1 chromatid entering the S phase. Coming out of the S phase, we still have two homologous chromosomes, but we've duplicated the DNA content. So each chromosome is now made up of 2 sister chromatids. And it's those 2 sister chromatids that will divide when we get to mitosis. And then the last phase is the G2 phase. And this is where we have the double checks and make sure everything has duplicated without error before we can proceed through mitosis. Okay, and then the small part here is mitosis, where we go through the stages of PMAT, right? The nuclear division. And then the nuclear division is followed by cytokinesis, which is when the cytoplasm divides. Okay, so this next slide is very much like what you would see with the onion root tip. So you'd cut off your onion root tip, and different cells throughout it would be in various phases. And so we have them pointed out for you here. You can see at the bottom one, metaphase, and you can see very much it's lined up in the middle of the cell. You see the earlier prophase slides, and interphase where the chromatin is not even condensed hardly at all. Anaphase is always so recognizable, where everything is super condensed and at opposite poles. And then even more, you see the furrow kind of forming in the middle on the slide, this telophase. And so we'll just look through the rest of these as if we were in lab and try to recognize the cells in the various stages of mitosis on some other slides. Here you have another stain for an onion root tip, and we can compare that with a whitefish blastula. So here, interphase, again, all the chromatin is pretty loose. Then we get into prophase, so we're starting to get everything ready here to start lining up for our metaphase plate, which comes next. So you can really see in that whitefish blastula, it's almost just like a stripe down the middle of the metaphase plate. And then in both slides, you see anaphase, where all the chromosomes are being pulled to opposite poles. And then in telophase, you can see where we're starting to form new nuclear membranes, and almost really, especially in the onion root tip, it looks like almost cytokinesis right there. Okay, and then on the right, next slide, we have other slides, more onion root tip with a different stain preparation in various stages of mitosis. And I think I'll leave it for you to go through these and see if you can recognize it on your own. And here's yet another preparation. And can you see which one is anaphase? I said anaphase is always the most recognizable. Again, here's our final slide, and you can see where we have interphase as the first slide, prophase as the second slide. The third slide at the top shows everything lining up in the center, the metaphase plate. Okay, the bottom row, we then have anaphase, where everything is pulled to the opposite spindles. And then we have telophase, where we're having our nucleus starting to reform around each new daughter cell or daughter nucleus. And then we have our final slide, which is cytokinesis, where we're basically forming our whole new daughter cells in the cytoplasm divides. And that's it. We're done with mitosis. I hope you feel like you understand it now.